Hi, my name is Philip Murray. Today I'll be discussing about our project about Coxsackie-Burnetii, particularly how it's a BSL-3 agent and the epidemiological signs and symptoms, extra precautions people should take when working with this organism inside a BSL-3 lab facility. Um, so one of the strains that's usually used is the 9-mile RSA493 strain, but generally Coxsackie-Burnetii is a gram-negative rod-shaped bacillus that produces spores, which is one of the reasons why it's classified as a selective agent because of the ability to produce spores and infect the respiratory tract of susceptible people. Speaking of susceptible people, susceptible populations include individuals that suffer from any chronic respiratory diseases as well as cardiovascular diseases because they can also affect certain arrhythmics of the heart as well. The route of transmission usually is through the respiratory route, but it can be ingested, is another way. But most routes of the infection come from aerosolized spores from farm animal feces or urine, usually by when farmers or workers are power washing the stables or they're sweeping up debris, and it can generate a lot of aerosols for you to increase that risk of inhaling it. Ingestion can also take place, like I mentioned before, but person-to-person transmission of if an infected person is infected with Coxsackie-Burnetii, it's very rare for that person to transfer via person to person. This agent is highly infectious, and in all it takes is about 15 organisms to actually cause an infection. If you are infected, some of the signs of this infection are high fever, myalgia, febrile illness within about 2 to 3 days, and then this can also form into atypical pneumonia, which include other symptoms of shortness of breath, coughing, and difficulty breathing. And then as well as it can affect the cardiovascular route, causing hepatitis, myocarditis, and pericarditis. And there are even neurological symptoms associated with infection that can cause encephalitis and other varying forms of encephalitis. However, if a person were to become infected with this, the treatment for this is usually about using 100 milligrams of dioxycycline per day over 2 weeks. But other antibiotics such as fluoroquines and minocyclines and TIM-P-SMX can also be used as alternatives. However, higher risks are for pregnant women too. If that's the case, TMP-SMX will be given over the course of the entire pregnancy until the 7-month mark. A whole cell formulation inactivated vaccine is available to those who are working with the agent. And it's highly recommended if you are doing research with this agent that you receive this vaccine. It's also labeled as QVAX, and it can be requested upon, and if you're working with the agent, it can be requested that you get that, and is recommended that you do get that if you work on this agent. When working with this agent, all cultures are completely done in a BSL-3 lab inside of a hood, and there's never work done outside of the hood. And lock boxes should be used when storing cultures of this. And however, if there is a spill or a contamination event inside the hood, it's important to fumigate the hood, completely close off, follow procedures. If it involves breaking, it involves spraying everything down with disinfectant and aerosolized formaldehyde or hydrogen peroxide to fumigate the hood and let it sit for about 2 hours. And then it's safe to wipe down everything again with 70% ethanol and then, I'm sorry, 10% bleach and then 70% ethanol. And that allows for that area to finally be disinfected. Going back to the at-risk population, individuals who work in the farm industry or live on farms are more subjected to increased risks of this bacterium due to the route of transmission of contaminated aerosolized animal feces going through the respiratory tract. Individuals who live or work on a farm or within a certain radius of a farm should think about, it's recommended that they get vaccinated, especially since, like I mentioned before, it can only take 15 bacterial cells to cause an infection. And those who are pregnant or becoming on pregnant also, and those who suffer from respiratory and chronic cardiac disease should also consider being vaccinated. especially if you live within a 4 mile radius of a farm, because there have been documented cases of spores actually traveling up to 3 to 4 miles from a farm into local suburbs or other neighboring areas. Um, One of the things that's really surprised me about this is the fact that um, one of the biggest things for biosafety is risk mitigation. So it's literally not just assuming that PPE is the um, most protective thing you can have. It's lower on the totem pole of the hierarchy. And the best defense is literally taking your experiments, addressing where there could be possibly events, contamination events, accidents, or any injury, and you take that out and consider doing alternative measures or alternative procedures that take away those risks. So, literally, the best defense of working with an agent like this, for instance, is to make sure that you are removing those risks of possible aerosolized contaminants, um, making sure you're not using any sort of sharps or things that could possibly cause sharps. And if you're going to use sharps, such as glass containers like CBT tubes, you're going to want to make sure that you follow even safer protocol, and you want to make sure if you're using glass tubes, you want to examine them before you take them. You want to make sure they're not cracked or anything, and you want to make sure you have the necessary equipment around to dispose of said sharps around, or else it could lead to potential accidents. But it definitely, the biggest case with this is making sure those risks are taken away, address those risks, eliminate those risks, write your protocols based off of performing things safely, and of course PPE and wearing respiratory protection, because since this pathogen can be spread through aerosolized routes and infect the respiratory tract. So never heard about this organism, period. Learned about it today, kind of a little worried about this kind of infection in general. I never knew this existed. And it allows me to take, this whole class has allowed me to address safety issues in my own current job, as well as risk mitigation and eliminating certain things that could potentially cause risk for not just my experiments, but other experiments as well.